So the topics that have been mentioned in the syllabus, they are Cnidaria, the polymorphism of Cnidaria, defensive structures of Cnidaria, their mechanism, coral reefs, their formation, metagenesis, and general features and life history of Obelia as well as Aurelia. In this lecture, we will discuss about the general features, classification, polymorphism, and cnidoblasts in particular, because the defensive structures, these are the cnidoblasts. So you can see the questions that have been asked in the past. In 2023 there was this question about the statocyst. We will discuss it later on. Then we have the question related to polymorphism. We have question related to polymorphism once again in 2017, 2021. So this is one important topic from this portion. So let's discuss that. What are the general features of the coelentrates of or the cnidaria? If you will see the shape of the coelentrate, if you have seen hydra is one of the famous examples from the coelentrates. You can see it has this body. The body has this outer covering called as the ectoderm, or the outer layer called as the ectoderm. The inner layer is called as the endoderm. In between ectoderm and endoderm, there is presence of mesoglea. There is a basal disc that is attached to the substratum, and inside you will find a gastrovascular cavity. At the same time, you will observe there are tentacles around the mouth. And one more thing that you will observe will be that the tentacles, they consist of the cnidoblasts. They have the defensive structures. Cnidoblasts, the defensive structures. So the cnidarians, they exist in two forms. One is the polyform and another one is the medusa form. Medusa is umbrella shaped. You can see here in this case, you can see this is umbrella shaped, and it has similar layers: ectoderm, endoderm, there is mesoglea, and there is gastrovascular cavity, and there is a ventral mouth, okay, with tentacles attached to it, and as well as gonads are present in this. So this is a general structure. If you discuss the features, the level of organization is tissue level of organization. The body exhibits the radial symmetry because it has one axis, and along the axis the body can be divided by any plane into two halves when this axis when the plane passes through the axis. So it has diploblastic germ layers. We saw there was ectoderm, where there was endoderm, and there was mesoglea. There was no mesoderm, so it is diploblastic. The habitat, it's mostly it's aquatic habitat with mostly marine and few are freshwater. For example, hydra is freshwater. Then the body form, it exhibits polymorphism. They can be trimorphic, they can be dimorphic, they can be polymorphic, and they show prominently two types of two forms, that is the polyp, which is sessile, and medusa, which is free swimming. Then the body cavity, it consists of a coelentron or the gastrovascular cavity. That's why the name coelentrata. So these cnidarians, they are also called as the coelentrata because of the presence of prominent coelentron. Coelentrata. If you will observe that these they have this body cavity that is coelentron. The coelom is acoelomate types, and they have special cells which are the stinging cells or the defensive structures called as a cnidoblasts. And mode of nutrition, it's extracellular as well as intracellular. These are carnivorous. They take the food from the ocean, from the sea, wherever they are present. They eat the carnivorous diet. Then the nervous system, they have a nerve net which has diffused nervous system, very diffused nerves. In case of reproduction, there is asexual reproduction by budding and fragmentation, and sexual using the gametes. We will study the life cycle, metagenesis. There you will understand the different types of reproduction in in the coelentrates, particularly in Obelia and Aurelia. Then the development is mostly indirect through the planula larva. So planula larva belongs to the coelentrates. Then respiration, it's by diffusion, and common examples include Hydra, Obelia, Physalia, Aurelia, sea anemone, then corals. These are the examples. If you see the classification, they have been classified into 4 different classes. First one is the Hydrozoa. Hydrozoa consists of polyp. So polyp is the vase-like structure. The vase-like structure is the polyp. So yes, this is polyp form. Hydrozoa exist in polyp form mostly. These are colonial or solitary. They show the alternation of generation. Alternation of generation is the change from sexual to asexual form. They exist in both polyp and medusa form, but the polyp form is dominant. Examples include Hydra, Obelia, Physalia, Velella. Then Scyphozoa is another class. Scyphozoa contains mostly medusa. They contain the jellyfish. You must have seen the structure of the jellyfish. It's umbrella like, okay? It has these tentacles. So Scyphozoa is mostly in the medusoid form, that is jellyfish form. They have a thick mesoglea. They are marine and the medusa form is dominant. Examples include Aurelia, Cyanea, Rhizostoma, okay? Then we have class Cubozoa. In case of Cubozoa, the dominant form is medusa and they have a box-shaped medusa. They have complex eyes and these are highly venomous. For example, Chironex. Then the Anthozoa. Anthozoa include polyps only. They do not have medusa stage and they help in the formation of coral reefs. Okay, for example, Metridium, Gorgonia, there is Madrepora. These are some of the Panatula. These are the examples of the Anthozoa. Okay. Then this polymorphism. I told you that there is something called as a polymorphism. As the word suggests, the polymorphism refers to the occurrence of multiple distinct morphological and functional forms. This is important. There is morphological as well as the functional forms. And this exists within the same species or the colony. If you will see in case of the cnidarians, these cnidarians, they have both the polyp and medusoid form. Okay. So there are various types of zooids. These zooids are the individual members of a cnidarian colony. Each zooid has a special and specific function. So due to polymorphism, there is this division of labor. They are genetically identical, but in the morphism, the form, the look and the work, that is different. Let's see what are the different types of zooids. First one is the polyp. Polyp is the feeding and asexual reproduction. It shows polyp is the feeding form. For example, Hydra, Obelia, these are sessile and tubular. Then you have Medusa. Medusa is a sexual reproduction showing zooid and it does the free swimming. For example, Aurelia, these are free swimming and these are umbrella shaped. Gastrozoid, gastric, gastro, it means the feeding. Feeding, it includes the Obelia and Physalia and they help in capturing and digestion of food. Then there is gonozoid. Gonozoid includes the presence of gonads. Gonads are from where the gametes are produced. For example, in Obelia, they produce the medusae and buds. Then dactylozoids. Dactylo is related to defense or the capturing of prey. It's found in Physalia. Physalia is the Portuguese man of war. It has nematocysts. Okay. Then nematophore. Nematophore is the free floating. It has float support and it has a gas float. So it is something like this and it has gas filled cavity inside. So it exists in Physalia and it keeps the colony floating. Let's see different types of these zooids. You can see in this case, this is Obelia and there is presence of, this is the gonozoid. This whole is the gonozoid. The medusae are produced from this blastostyle. This is called as the blastostyle. Now this portion is the gastrozoid. This is the gastrozoid. You can see there are tentacles. From here the food will enter into the body. So this is the gastrozoid and it is attached to the main body that has the coenosarc and the perisarc. So this is the gastrovascular cavity. we will discuss it in detail in Obelia. Then you can see Medusa. Medusa is umbrella shaped. You can see it's umbrella shaped and it consists of gonads. It consists of gonads. It is sexually reproducing form. You can see there is presence of manubrium. Manubrium, from where the food enters. It is the pharynx like structure, mouth like structure. There are gonads present. There is endoderm, epiderm. It has this ectoderm, then mesoglea. So this is the structure of Medusa in general. Let's see some different zoids. As I told you that there is this gonozoid from, or the gonangium, from where the Medusa arise. There is presence of mouth and tentacles in gastrozoid. You can see there is tentaculozoid. Tentaculozoid will act as a tentacle and it also will contain the nidocils. There is this dactylozoid. Dactylozoid is for defensive structure. There is this gonozoid. There is skeletozoid, which acts as a skeletal element. There is hydrorhiza. Hydrorhiza is the main part of this, from where the zoids arise. So it's like a root. Then you can see here, this is pneumatophore. This is pneumatophore, the gas filled, found in Physalia. It also consists gonozoids and gastrozoids and dactylozoids. So these are the different zoids. At the same time, you can see in this structure too, you will see there is presence of dactylozoids. This is a dactylozoid, this is the gastrozoid, this is the gonozoid, this is the phylozoid. So there are different types of zoids. So you can make any of the diagrams in case of polymorphism to discuss the polymorphism. Now one thing is there, that what is the importance of polymorphism, why these organisms show the polymorphism. Okay, let's discuss. First of all, the significance. First of all, you need to understand that the polymorphic forms, they help in the division of labor. They help in the specialization. There are special zoids doing special functions. Then you have to know that it also helps in adaptation to changing environment. It helps in increasing genetic diversity. Because there is this polymorphic forms, there are different forms. So yes, there is increased genetic diversity. Then you will see there is reduced intraspecific competition. Within the species, there is less competition. There is enhanced predator-prey dynamics. There is maintenance of biodiversity due to the interaction. It also acts as a genetic basis for evolutionary changes. So these are different significances of the presence of polymorphism. Now there are also some theories related to polymorphism. You can mention some few of the theories. There are some theories related to the same. First theory is the polyorgan theory. So it says that there are these different zoids, presence of these different zoids. These different zoids, they act like polyorgans of the colony, thus exhibiting the division of labor. Second theory is the medusome theory. The medusome theory. It actually, this theory, this medusome theory, it says that there is this hypothetical medusome theory that focuses on the medusa stage of the colonial hydrozoans, and propose that the polymorphism within this stage, they play a significant role in shaping the organization and functioning of the entire colony. So the medusa stage shows polymorphism, which is extremely significant for the colony. It helps in adaptive significance. It has adaptive significance. Adaptive significance meaning that it helps in the functional integration of the body. Then third theory is polyperson theory. According to this theory, each zoid has its own individuality. And they are similar to societies. They interact to work together. So this is about the different theories related to the polymorphism, and these are the different forms and diagrams you will make in the exam and also write the significance. So this is about the polymorphism. Now next we will discuss about the cnidoblast or cnidocyll or nematocyst. This cnidocyll, it is a specialized and unique. It is unique to phylum Cnidaria. That's why it's called as Cnidaria, because it has nematocyst. These are specialized, okay? These are specialized cells and they are unique to the phylum Cnidaria. At the same time, these organelles, they are called as a nematocyst. They help in the defense, prey capture and attachment. They have hypnotoxin in it. So the structure include that there is this nematocyst, okay? You will see there is this nematocyst capsule. This is opened one. When it's closed, it remains inside like this. So it has operculum. This is the operculum. This is everted shaft. This is everted shaft. These are the stylets, okay? These are the stylets. These are the spines. And then you will have a base of the tube. So this is the base of the tube and the tube extends. So, or the thread. So it has an operculum, that is the lid-like structure. Cnidocyll, it is a hair-like trigger that detects the stimuli. Okay, there the cnidocyll will be hair, which will act as a small hair-like structure. Okay, this is cnidocyll. It detects. Okay, it detects the stimuli. Then there is coiled thread or tube. It's harpoon-like or tube-like. It has toxins sometimes, and there is capsule where the nematocyst lies inside. Okay, then you will see there are different types of nematocysts. First one is the penetrant. They are for penetrating, they are for stinging and injecting the toxin. They have barbs on it. It's found in Hydra and Physalia. If you will see the diagram, it's something like this. For example, here you have the, and it will be something like this. It will have barbs on it. So this is the penetrant type. Next is the glutinant. Glutinant is helpful for the addition. Okay, it is helpful for the addition. Addition means it is for attachment. So this is the glutinant. It is for attachment, addition. It has sticky thread which entangles the prey, for example in sea anemone. Then we have volvent type, volvent or desmoneme type. It helps in the entangling. For example, this is the nematocyst. It helps in the entangling. Here is your twig or prey, whatever it is. So this is the volvent type. After the volvent, we have stereoline. Stereoline is helpful in the detection. It is helpful in sensing and defense or defense. It's rare type and it's used for sensing the mechanical stimuli. So it is, in general, it's like this. It will be small barbs. So this is the stereoline. Then we have a lasso type. Lasso type, it helps in coiling around the prey. Okay, it has this lasso, so coiling around the prey. So these are different types of the nematocysts. Now there is this mechanism. How does it activate? How does it open up? How does it work? So these nematocysts, you will see that in normal conditions, it remains closed. It remains coiled inside the nematocyst. Okay, they remain closed in the capsule of the nematocyst. When any of the pressure is activated, any pressure is occupied, any pressure is exerted on the nidocyle, on the trigger, what happens? It will release its stylet and it will release its shaft, and then it will push it outwards, and then it will either capture the prey or inject the hypnotoxin or just capture whatever or entangle whatever it is going to entangle. So the molecular mechanism includes that first of all there is stimulus detection that activates the whole structure. There can be mechanical or the chemical stimuli. It activates the nidocyle. As I said that there is a trigger here. Okay, there is a trigger like structure, nidocyle. Then what happens that it will lead to the membrane depolarization, and it causes the calcium ions to release from the internal stores into the cytoplasm. So calcium will enter into the cytoplasm. This portion is the cytoplasm. Okay, so this portion is the cytoplasm. The calcium will enter into the cytoplasm. So what happens that calcium will enter into the cytoplasm after the mechanical stress is exerted, then or the chemical stress. Then after the ion movement, it will also trigger the water influx via the osmosis, and this will include the rapid movement of water into the inflow into the capsule. It will increase the hydrostatic pressure, so there will be increase of water supply into the capsule. Once the water is supplied into the capsule, it will help in the explosive eversion. Internal pressure will be high and the operculum will open. The operculum is the lid-like structure. It will open as the internal pressure increases, and then the barbs, they will penetrate the tissue of the prey or victim, whatever there will be presence of any predator. It will push the barbules into the body. Then the toxins, neurotoxins or porins, they are released, and the molecules which are included, first one, the calcium is there for trigger, and polygamma glutamate. It holds the negative charge and it draws water. Then nidoin, that is strengthens the capsule wall, and there are toxins which are neurotoxin or porins, so they paralyze the prey. Overall, you know this mechanism now, and the functions include the simple function of capturing the prey or defense or attachment. So this is the function. Locomotion is also possible through it. It's been asked and it has been repeated many a times. You will see that the coral reefs, it's one of the favorite topics of the UPSC in civil services as well as in the forest services. Like you have the questions related to give an account of coral reefs and their constituent compartments and significance, the corals and coral reefs. So this is the theme that has been repeated many a time. So the topic of coral reefs is important. We will see the structure and types of coral reefs and also the theories related. Then the topic of metagenesis, that includes the change of the cycles, the sexual and asexual cycles. We will study it and then discuss it in Obelia, as well as we will see what are the different phases these organisms go through during the metagenesis. So starting with the corals. In this map, you can see the distribution of corals across the globe. You will see that it is across the shores, it is across the lines. We will see the shelves of the continents. It is present across the shores. So the maximum density you will observe across this area, which also contains the biggest and the largest coral reef, that is known as the Great Barrier Reef. So you can draw this map while answering the question about corals. So this is a general distribution of coral reefs. Now I'll let you know the structure of one corallium. So these whole colonies of coral reefs, they contain lots of corals, and the structure of single coral, let's see that. This is called as a corallium. It has a basal plate. At the base there is a basal plate. Then there is theca. Theca is the covering. Theca is the outer covering. Okay, theca is the outer covering. Then there is inside, you will observe there is stomach, that we already called that as the coelentron. Okay, it is called as the coelentron. You will see there is gastrodermis. Gastrodermis is the layer that surrounds the coelentron. Then again you will see the digestive filaments inside it. There are digestive filaments. There is presence of a septum. There is presence of mesoglea. Mesoglea is present. Then there is cenosac. Cenosac is the layer present over the gastrodermis. So there is cenosac. There are presence of tentacles. There are nematocysts. There is outer epidermis. There is this mouth, and then it goes into the coelentron, that is the stomach. So this is the basic structure of a corallium. Now we need to understand that how the corals are formed, the formation of coral, and at the molecular level what happens. First of all, you need to know that there is presence of zooxanthellae. Okay, zooxanthellae are the symbiotic algae. Symbiotic algae means it gives benefit to the corals, and the coral in turn gives benefit to the zooxanthellae. So zooxanthellae, what they do, they live inside the coral cells and they perform the photosynthesis. Okay, they perform the photosynthesis. Thus they provide energy rich compounds. So they provide food. So zooxanthellae provide the food to the coral. And in turn the coral, it provides the shelter to the zooxanthellae because they live in this. Then the photosynthesis, it's by zooxanthellae produce the organic molecules like glucose, glycerol, amino acids, and this energy, it's used by the corals in order to form the skeleton. So the energy produced by the zooxanthellae, it's used by the corals to form the coral reef. Now the calcium ions and bicarbonate ions. You know the seawater, it contains minerals, so it contains calcium as well as bicarbonate. The oceans, they act as the largest sink of the carbon dioxide. So when this carbon dioxide, they interact with the water molecules, they form the carbonic acid, and from there the bicarbonate ions are released. Calcium is present in the seawater. So these specialized transport proteins in the coral epithelium, they help in the calcification. It There is the formation of this calcifying fluid. Okay, there is formation of this calcifying fluid. There is formation of this calcifying fluid. Once this calcifying fluid is formed, then just give me one moment. Okay, so this calcifying fluid is formed beneath the polyp, and this calcifying fluid, this in turn helps in the formation of the calcium based structure. So what happens, as I said, carbon dioxide is involved in the formation of the carbonate acid. This carbonic anhydrase enzyme acts on this to form the bicarbonate ion. Then this helps in the steady supply of the carbonate ions. There is need of continuous supply of the carbonate ions in order in order to form the calcium carbonate. Now we have calcium ions and we have bicarbonate ions. When they combine, they form the calcium carbonate. Okay, there is this reaction that helps in the formation of calcium carbonate, solid calcium carbonate, and it's in the aragonite form. Now this molecular structure, that is crystal based crystal structure, it forms as a small nucleation site, and around this the whole corals further deposition of the calcium carbonate takes place, and this coral, this increases in size, and there is formation of this small nucleation site. So there is this small nucleation site, and around the nucleation site whole coral reef is formed. Then this coral skeleton, there is this hard exoskeleton of coral polyp. If you will see small corals, it has a hard structure outside, and it multiplies, and continuously calcium carbonate is deposited over the 1,000 years, and there is formation of this reef. So this is how the coral reef forms. Okay, then there is the topic of types of corals. There are different types of corals. Okay, first one is the fringing reef. If you will see in the diagram, you will observe that fringing reef, these are directly attached to the shore, and they border a coastline with a narrow lagoon or no lagoon. It's not important that they have a lagoon between reef and land. And it is common along the volcanic islands and continental coast. For example, coral reefs in the Red Sea and Hawaiian areas. Okay, so if you will observe, for example, this is the volcanic island. Then you will observe that there are small these fringing reefs around it is closer to the shore. It is closer to the shore of this volcanic island or a small island. So this is the fringing reef. It's found in Hawaii, it's found in Red Sea. Next one is the barrier reef. So barrier reefs, these are separated from the mainland or island by a wider or deeper lagoon. Example is the Great Barrier Reef in Australia. If you will see, there is an island, but at the same time you will see there is different distance between the coral reef formed over the time and the main island. Then we have atolls, and atolls are very famous in Lakshadweep and Maldives. You will see that the main island, it has completely subsided into the ocean. It has completely gone into the ocean, and it has left a big, large circular structure made up of corals, which is called as a atoll. It's ring shaped and it encircles a lagoon. So inside the atoll, there is presence of this lagoon. For example, in case of the Maldives and Bikini Atoll. Then we have patch reefs. Okay, there are certain other types of reefs which are not much discussed. These patch reefs, they are small and isolated reefs, and they grow from the bottom of the island platform or the continental shelf. So it will be something like this. For example, this is the continental shelf. Okay, this is the continental shelf, and it will grow somewhere like this, and this is the ocean. So it grows somewhere along the shelf and it forms some structure like this. So these are patch reefs. These are found between the fringing or the barrier reefs. Then we have platform reefs. These are also called as the bank reefs. These have flat topped reefs and they are also found across the continental shelves. There are ribbon reefs. These reefs, they have winged shape. Okay, they have winged shape. They have winged shape like this, and they are part of the barrier reef system. For example, in northern area of the Great Barrier Reef. Then micro atolls, these are smaller vertical growth due to a sea level. They have a limited vertical growth due to the sea level. Okay, and they have dead upper surface and living coral around the edges. There is dead surface, but there are living corals across the edges, and it will help in formation of deposition and growing of the coral over time. So this is the these were the types of the corals. Now we will discuss about the significance of corals. The corals, they are very significant as in because they have ecological significance. As you can see, they have ecological significance. They have um, uh, environmental significance. They have economic significance. They have scientific significance. They have educational significance. First of all, the ecological significance. They act as a biodiversity hotspot. You know that zoos and there are many organisms. If you have seen the movie Nemo, you will observe that that fish is living in those moving structures. They are actually the corals. So these act as a biodiversity hotspot. These are also called as these are the forests of the ocean. So 25% of all the marine life, they, despite covering less than 1% of the ocean floor, they support all this 25% of the marine life. They act as a nurseries for the marine species, like crucial breeding, nursery grounds for fish and invertebrates. Then primary productivity, they have high productive ecosystems. Zooxanthellae support food chains via the photosynthesis. Then environmental significance, like coastal protection, there is carbon and nitrogen cycling, water quality maintenance. So these are all the qualities and ecological significance of the coral reefs. Then economic significance, it includes that fisheries, tourism and recreation, medicinal resources. There are scientific and educational significance, like natural laboratory. It acts as a natural laboratory. It acts as a climate indicator. So if the damage to corals happen, it is due to certain reasons. Okay, first reason is the climate change. There are certain reasons for damage to the coral reefs. First one is the climate change. There is coral bleaching due to rising sea temperature. Ocean acidification, it also causes the coral bleaching. Then pollution is caused by sewage, agriculture, plastic and other harmful substances. Sedimentation occurs. Sedimentation means that the sediments from the forest, from the coastal areas, it comes on the and gets deposited on the coral reefs. Overfishing causes a problem. The destructive fishing, coastal development, even coral diseases are there. There are certain bacterial, viral, fungal diseases. There is pressure of tourism. Invasive species cause problem. So these are the issues that the coral reefs face. Then there is this theory of coral formation. First of all, we have Darwin's subsidence theory. According to this theory, what discussing about the theories of coral formation. First theory is the Darwin subsidence theory. What happened that there was an island, and the coral reef, it started to form along a shore, on the shores of the island, and with time this island it's subsided within the water, and the reef, it comes to the surface of the water. So the subsidence theory says that reefs begin as a fringing structure around the volcanic island. As these islands gradually sink, the reefs grow upward to maintain the position near the surface, transitioning into a barrier reef and ultimately atolls once the island is fully submerged. Then the coral drilling has confirmed coral over volcanic rock beneath the atolls, and this is a supporting evidence. This is a model for the Darwin subsidence theory. Then we have Daly's glacial control theory. According to this theory, what happened across the ages, as the temperature rose, the sea level drops during the ice ages. This is what happens during the cycles, Milankovitch cycles. The temperature rises at one point in the cycle, the temperature falls. So during the time when the temperature falls, these are the ice ages. So during the ice ages, they get exposed. There are these platforms which get exposed, and when the sea level rises post glaciation, corals colonize these platforms and they start to grow upwards. So water comes down when there is glaciation, and over these platforms the corals form. When the water level goes up, over these platforms these corals are formed and they are observed above the water surface. The evidence that supports this theory is the consistency in the lagoon depth, the plateau structures underlying the reefs, and timing aligning with the glacial cycles. So these cycles, these are the Milankovitch cycles. If you will Google it up, you will see there is a term, Milankovitch cycles, and according to these cycles, the glacial cycles are explained. Then another theory is the banks and standstill theories. According to this, the bank theory, it was given by Murray. So the bank theory, it is regarding the submerged banks and Murray's. The coral reefs, they grow on pre-existing submerged banks. Okay, so there are these coral reefs, they start growing on the continental shelves or the ridges without the need for the subsiding of the volcanic island. So there is already one platform or submerged bank, and on this submerged bank slowly the coral reefs grow. According to standstill theory, the coral growth occurs on the static platform. Reef structure depends on the platform's shape at the time of the colonization. So these theories explain the reefs in areas without the volcanic activity or subsidence. So these are the theories regarding the coral formation. There is not need to explain them if only the corals are the question regarding the corals come in 10 marker, but in 15 and 20 marker question, there you can add these theories. So this is all about the coral reefs and formation and some theories. Let's now discuss the topic of metagenesis. Okay, so metagenesis in cnidarians such as jellyfish or jellyfish which include the Medusozoa, Hydrozoa, Scyphozoa, Cubozoa, Staurozoa. It is the regular cycling between the asexually reproducing polyp and sexually reproducing medusa. So the polyp form, which is the vase-like form, it develops asexually. For example, it develops by budding. So what happens that the polyp that reproduces asexually, they transform into the medusa. They give rise to medusa, which is a sexual form. So this transition between the asexual and sexual form is called as the metagenesis. So there are stages. First of all, you need to know that there are two stages. One is the polyp and medusa. Polyp is stationary, cylindrical, and it reproduces asexually. Then medusa is the free swimming bell shaped and it reproduces sexually. So you can see the metagenesis in Obelia. The medusa, it is umbrella shaped and it's sexually reproducing. You can see there is formation of sexual reproduction. Ephyra is formed, then there is zygote formation. Zygote gives rise to planula larva. Then there is polyp. This polyp gives rise to this polyp stage. It gives asexual reproduction. It produces the ephyra, which is the juvenile medusa. This juvenile medusa grows into medusa. It will be more clear in the Obelia, the topic of Obelia. So the life cycle, first there is this polyp stage that attaches to the surface and it often forms the colony as an Obelia. It reproduces asexually through budding, and in some jellyfish strobulation takes place. And then segments are formed, and these segments they develop into medusae. In medusa stage, this medusa, it detaches from the polyp. You can see this is this detachment. This medusa, it detaches from the polyp, and then this is equipped with the bell, tentacles, gonads, and they release eggs and sperm into the water for external fertilization. There is this planula larva. There is a motile ciliated larva that settles on the surface and it develops into a new polyp, continuing this cycle. So there is a larval stage, that is the planula larva. So alternation in generation in Aurelia, it is also called as metagenesis, and it's the phenomenon in which two different types of body forms, asexual and sexual medusa, they alternate in the life cycle of an organism. In Aurelia, that is the moon jelly, the alternation is between the sessile polyp and free swimming medusa stage. So this is about the metagenesis. Then how metagenesis shaped the cnidarian evolution. Over the time, you will observe that there was the polyp baseline, which was the basic ancestral cnidarian, and it was stationary, but it had to disperse, it had to reproduce, it had to spread, it had to capture prey. So over time, what happened, medusa was an innovation in the evolution. It was an evolutionary event within the Medusozoa. It gave rise to the motile medusa, enabling the broader dispersal and sexual reproduction. There was lineage specific losses, such as some groups they retained both the stages of medusa and polyp, and in some stages one or the other form was dropped. Then gene level evolution, there were new and conserved gene families which drove the development of the complex medusa traits, and there were also adaptive successes like with alternating cycles. It added the resilience and versatility, helping the cnidarians thrive across the diverse marine environments. So this is about the metagenesis. We have discussed about the coral reefs. Now after this we will discuss about the Obelia